So if we have any questions, please, now's a great time to.
So how do you, I'm talking now about the molecular-oriented graphs for a moment. How do you see these new sorts of techniques being used for either the questions that we currently ask, or what questions do they allow? Do you see these tools potentially opening up? If they were available in Patrick, if a tool that was based on this would pop up in Patrick, how do you envision that they might be used by a biologist?
Okay, so the ad hoc use is folding and inverse folding of RNA sequences. And by that I mean, if you're interested in a short RNA interacting either with DNA or with another RNA, these tools can give you configurations, contact configurations of the RNA binding, and gives you structural understanding, but no spatial embedding. So none of these algorithms are really referring to a lattice or to some 3D embedding. Currently not, but they give you contact information. You know who's paired with whom. So this is one class, and this is the folding. This is a mapping from the sequences to the structures. There is a completely different inverted applications line, which is inverse folding. Say you have a certain shape of an RNA, and you want to create sequences that are likely to produce that shape. Or, in fact, you want to manipulate the shape of that RNA, and you want sequences that have a certain structural feature you want. Then these tools can actually compute the inverse fold, which is equally complicated but inverse. The inverse fold can create subsamples of potential candidates of these sequences efficiently. These are also polynomial time algorithms, and those are ad hoc applications for the molecular foldings.
How about predicting something that would bind to that RNA and knock out its function?
You mean a molecular binding, like having a short sequence of an RNA and like some kind of antisense type of binding? Yes, this is very much possible. And it's embarrassing enough. We have done this in a math department. We have barely been able actually to find the time to do the conceptual coding. So right now we push the coding to a form that this is more user-friendly and things like this, because it's a long path to make things interesting and applicable for you guys, right? So we're in the process of doing this right now, and I'm quite excited that we have a chance actually to also hear what it is we can do in order to make this more useful, right? Because they were basically designed to get a publication done. We needed to have a C code that would compile and run and produce the data we needed in order to show specificity and PPV values, but there's much more to those tools. So they have not been properly PR'd.
My thoughts are, if you can knock out the function of a ribosomal RNA, you can affect pathogenicity of the organism.
Right. Right. So I think they're fairly powerful, and I think if you think, I don't even know whether they're thinking outside the box, but if you think of function, the patterns that you're actually looking for are entirely different. Let me try to make that point. If you go into this microcosmos where one RNA has a whole network, one structure has a whole network of sequences that fold that structure. Right? So there's an exponential number of sequences, all of which form this particular structure. And I can find very easily two sequences where not one nucleotide in any position is equal. They're completely antipodes. And they still make the same structure. But many of the searches are actually sequence based. So I think there's a real possibility to shift this to this functional genomics paradigm, where you actually look for patterns, but the patterns are given by the structure. And these folding algorithms are, of course, very important here. Is this clear? Or?
So what you said is that looking at the sequence alone, you can't find these properties.
Exactly.
Only looking at the sequence with respect to its functionality can you,
Correct.
and in this case, it's shape functionality.
Correct.
Can you understand their equivalence? They're equivalent under shape.
Exactly.
They're not equivalent under
Correct.
sequence.
They're part of the same neutral network.
So that's what a neutral network is.
But as a biologist, I'm having a tough time to wrap around sequence context to that shape, right? So for example, if you take, like, say, small RNA regulation of RNA, transcriptional regulation of RNA and RNA, so how do I go from that? Because I know that a certain microRNA, because of the sequence context, it's going to go and bind to the 3' region or whatever it is for a mRNA and regulate whether a protein is going to be translated or not. So how do I go from that sequence context or that sequence specificity to that shape?
So I'm not trying to say that sequence content can be totally ignored. What I'm saying is that, of course, molecular interactions have a very clear sequence signature. Some of them are totally dominated by antisense binding, right? It has to be the exact sequence of nucleotides, and otherwise it will not work. But what I'm also saying, if you look at the normal transfer RNA, there are bazillions of sequences that are not coinciding in a single nucleotide that will fold this transfer RNA.
It's not clear. I mean, a hypothesis would be that any of those sequences, the ones that fold the same, would produce the same protein.
So they're structurally, on a
That's a
certain level, on a
hypothesis.
certain coarse-grained level, they're structurally equivalent. For the particular molecular mechanism, you may need this particular, short sequence of nucleotides to be present to make this work. I'm saying there are two sides to it, and we've been focusing a lot on the sequence level. And I'm trying to stress the point that there is a completely different level here, namely that you have this kind of more abstract patterns. They're not sequence, they're still patterns. You just have to generalize the concept of patterns a little bit.
So I have another question.
Yeah?
So, and this has to do with the more general network. So in the particular case you brought up was SIR, but so SIR is a kind of a reaction diffusion process over a network. But so the more generally interesting question is reaction diffusion. It could be spin glass style computations. It could be all kinds of different things over networks. And so do you think that this sort of approach, and obviously this is relevant for learning and adaptation and computation of things. So do you see these techniques as being relevant, quite generally, not just with the spread of the flu or something, but just dynamics over these networks?
Right. So this is multi-layered answer, right? On the one level, right? The RNAs are for me particular networks. That's really what they are, because I tend to draw them as diagrams. I put the sequence on the x-axis and then I draw the arcs in the upper half plane. I'm German, right? I like to organize things. And so they're a network for me. But they're only particular classes of networks. So what I'm really interested in are interaction networks. And I want to understand interaction networks. And what this group is going to do is going to extract structural principles that allow me to describe processes over those networks in any way that makes sense. Right? And that I can guarantee you we will do. We are not going to confine ourselves to something that I particularly well do, or he particularly well does, or Mike. Because Mike is also going to learn new things with us if it is required. So this I can guarantee you, and it's not going to be anything that is not completely tailored to the particular problem. But what we really try to understand are interaction networks, not even SIR. right? This is something that we've done over the last 1.5 months together with Andre, and we were kind of and discussing with Stephen and learning a lot from Madhav and Anil. So we thought that's a cool thing to get started and to make some progress, and we're kind of puzzled about the structure that we see right there already. So we're quite excited about the first interactions that have been going on, and come my molecular guys. I hope to see more of you to talk to us, and then we make it happen and get these folding algorithms maybe put to use by you in a meaningful way. I would be quite happy about that.
Great. Well, thank you very much for a very informative question and answer session and talk, and we very much appreciated.